Welcome, everyone. I'm Nancy Maizels, chair of the International Scientific Council of the Israel Cancer Research Foundation, or ICRF. ICRF has been working for nearly 50 years to support the very best cancer research in Israel. Since its founding in 1972, ICRF has given out about 2,500 grants, totaling nearly $80 million to outstanding investigators at the leading universities and research institutes throughout the land of Israel. And in this year, we're currently supporting about 70 investigators with about $5 million a year in financial support. This is the 9th in a series of webinars that we began last spring for a truly wonderful audience who's shown themselves really interested in learning more about human health and disease at this time when we're facing extraordinary and unanticipated challenges accompanied by a real deluge of information. The focus of these webinars is targeting cancer with new therapies. And to help those of you who don't remember or maybe never even learned some basics of biomedical research, and we appreciate those people are rare in our audience, we're now putting a glossary up on the website with a little bit of background information on each talk and a few terms that will help you understand the talks better. And this knowledge will accumulate as you go from talk to talk. So you'll be really gaining your expertise in very contemporary and important issues in human health. So today we're going to talk about two kinds of ways of cancers that we know about quite well. About 20% of human cancers are caused by viruses. Vaccines, as I think we all know, teach the immune system to recognize viruses as novel invaders and target them for destruction. And we're very much aware right now how powerfully vaccines can work to fight infections. A striking success story in this regard is a vaccine against cervical cancer. Cervical cancer used to be a major public health problem, but a lot of research enabled development of a vaccine that was approved in the last decade or so. And Sue King and Mark Israel will tell you about that, targeting cancer with a vaccine. But what about the 80% of cancers that aren't caused by virus infection? That's most cancers. How can we identify them and nip them in the bud? As I hope everyone remembers or will learn quickly, chromosomes contain the genetic material of the organism. If you don't remember this, look on the ICRF website, or better yet, look at my favorite textbook, the free online Wikipedia, to learn more about what a chromosome is. Normal human cells contain 46 chromosomes, but cancer cells frequently contain abnormal numbers of chromosomes. This is referred to as aneuploidy. And this is caused by extra copies of some chromosome, too few of others, contributing to a cell's rapid division and taking over the body. We'll hear in the second part of the webinar from Uri Ben David at Tel Aviv University, whose lab has shown that aneuploidy may be the Achilles heel of some tumors, and that with a little bit of outside prompting, it may be possible to stop aneuploid cells from dividing and thus stop cancer. So first, I'd like to turn to Mark Israel and to Sue King with a little bit of introduction. Sue King is a board member, and she leads the Women's Advisory Council of ICRF. Sue is a former investment banker and asset management executive, and she currently works in private equity and venture capital. Dr. Mark Israel is the Emeritus National Executive Director of ICRF and Preston and Virginia Kelsey Distinguished Chair in Cancer at Dartmouth Medical School Emeritus and Director Emeritus of Dartmouth's Norton Cotton Cancer Center. So Mark and Sue.
Happy New Year, Mark. It's so nice to see you.
Well, it's a pleasure to be here with you today.
Terrific. Well, we're going to keep you tied to ICRF as long as we possibly can. But I'd love to ask you a few questions as part of today's Brilliant Minds webinar. Deaths from cancer in both the US and in Israel far outstrip the number of deaths from COVID-19. And the issue of vaccines to prevent the COVID infection has brought great attention to ongoing work to develop vaccines to prevent cancer. Now, while most of our listeners will be familiar with something like the polio vaccine or other vaccines against viral and bacterial illnesses, are there currently any vaccines that are effective in preventing cancer?
Sue, yes, there are. The study of vaccines to prevent cancer has been ongoing for decades, but the development of a successful vaccine against cancers caused by the family of viruses called human papillomavirus has drawn attention to vaccines as an effective preventive strategy against several different cancers caused by papillomavirus, including such cancers as cervical and penile cancers.
That's so interesting. Can you tell us about the vaccine that's used to prevent cancers caused by HPV?
Well, it's not widely known, but probably between 15 and 20% of all cancers are known to be caused by infectious agents. So cancers such as some liver cancers, and we mentioned cervical and other kinds of cancers. Those that are caused by HPV, human papillomavirus, are effectively treated with a now second generation vaccine called Gardasil. The first version of Gardasil was effective against a smaller number of different human papillomaviruses, all of which could cause a variety of cancers. And in fact, of the types of cancers, such as cervical cancer, which are caused by HPV, approximately 90% of those tumors result from HPV infection. And so both the first and second version of Gardasil, the second version, which is effective against a wider number of different human papillomavirus strains, has been found to be both highly efficacious and without significant side effects. And much like the COVID vaccines, these vaccines are directed against structures on the surface of those viruses.
Will it be possible to develop vaccines to prevent tumors that are not caused by viruses?
Well, I certainly hope so. Vaccines work by stimulating the immune system to recognize foreign structures, novel structures, structures that the organism, in this case humans, has not grown up with from the time of conception till it's a fully formed and ultimately a mature and functioning individual. Typically, these vaccines then recognize foreign as opposed to self or normal structures in the body. And that recognition, and in this case, it would be by vaccines, is the first step in stimulating an immune response. And so if cancers were to have novel structures, which we know they do, it should be possible to develop vaccines.
Thank you. What is it that leads to the development of novel molecules on the surface of cells?
Well, cancers are the result of either altered genes or alterations in the regulation of genes. That is to say, either genes have been structurally damaged and they're altered and they are incorrectly repaired, and that gives rise to a similar but slightly different molecule than the normal corresponding gene would give rise to. Or the gene is inappropriately regulated in the cancer cell. In other words, there are many genes that are turned on during fetal development that the adult organism doesn't need. And so either genes which should be silenced at a particular time or should be silenced in a particular location. I mean, we don't want liver genes turned on in the lung, and we don't want lung genes turned on in the liver. So either altered genes or altered regulation of genes can be recognized as the body can be recognized by the body as novel. And the body can then initiate a immune response against what appears to be foreign invasion. Now, there also is another way in which alterations in expression occurs, and that is as a result of changes in the number of chromosomes. When one cancer cell gives rise to two cancer cells, the normal expectation is that the genes in the original cancer cell are duplicated and then equally divided into two tumor cells. But in point of fact, sometimes they aren't divided equally, and you get the wrong number of cells in each of the two daughter cells. And that condition is called aneuploidy. And it results in too many chromosomes, which are packets of genes, in one cell and too few chromosomes in the other daughter cell. And that leads to the inappropriate expression of genes, and that emerges as a novel target for the immune system. And indeed, aneuploidy, the incorrect number of chromosomes in daughter cells of any given cancer cell, will be the topic today by Dr. Uri Ben David, who studies aneuploidy in work supported by the Israel Cancer Research Fund, and we're going to hear from him during this program.
Wonderful. Well, thank you so much for your time, Dr. Israel.
My pleasure. Nice to see you, Susan.
So Mark and Sue, thank you very much for reminding us of how effective vaccines can be against some kinds of cancer that are caused by viruses. That's a wonderful and very recent accomplishment. Now I'd like to turn to accomplishments that are coming up and that should change the way we treat cancer in the near future. And it's my pleasure now to introduce Uri Ben David from Tel Aviv University, who is a new junior faculty member there. He started his lab at Tel Aviv University in the fall of 2019. He got his undergraduate degree at the Hebrew University of Jerusalem, working on aneuploidy way back then with Nissim Ben-Benisti. He has then proceeded to a postdoc in Boston with Todd Golub at the Harvard Broad Institute. Throughout his career, he's received some wonderful awards, including the Dan David Scholarship, which is an international and interdisciplinary award that's very, very prestigious as a graduate student. And then his postdoctoral work was supported by EMBO, Rothschild, and Human Frontiers Science Program fellowships. After his arrival in Tel Aviv, he's received the American Association for Cancer Research Next Generation Star Award, and he's also received an Israel Gesher Award from the ICRF. Uri's research focuses on a genetic malady known as aneuploidy that's found in many solid tumors, and Uri will tell you more about what aneuploidy is and how his goal of cracking cancer aneuploidy to inhibit cancer growth. So Uri, welcome. It's wonderful to see you.
Thank you, Nancy. It's wonderful to be here.
Thank you. I'd like to find out a little bit about why you became interested in science. I'm always very curious about this.
Well, I actually became interested in science during high school. I took chemistry, it was my major, but my real interest in science actually did not start from science, but from the scientific method. And it actually happened when I was an intelligence officer at the IDF. Because, you know, what do intelligence officers do? They try to gather information and build a picture of the reality with missing and partial pieces of information. And then they try to identify gaps in information and follow these gaps, try to answer these questions and every answer basically leads to another question and so on. And this is actually very similar to how we do science, with one big difference, which is in intelligence, the whole idea is to keep the information to yourself and to prevent your enemies from having this same information. Whereas in science, the whole idea is to share the information for the benefit of human health. So right after my service as an officer in the military, I decided that I actually wanted to do science. And so I decided to study biology for my undergraduate studies at the Hebrew University. And that's when I really got interested in genetics and later on in cancer research.
So that's a really wonderful story. Thank you for telling everyone that. And I hope it inspires some other people to go into science or to try to learn more about science. Now I'd love to hear about the research you're conducting in your new lab with your Gesher Award from ICRF.
Yes, I will happily tell you about it. So I will share my presentation with you all now. Okay, so the goal of cancer research is basically to kill cancer cells. Now, it's actually very easy to kill cancer cells. You can just pour bleach on them and they will die. The challenge, of course, is to kill the cancer cells without killing or hurting the normal cells of our body. So it needs to be selective killing. In my lab, we're trying to achieve this goal by targeting a unique trait of cancer, its abnormal number of chromosomes. Changes in the chromosomal composition of the cell, also known as aneuploidy, make for a fundamental difference between normal and cancer cells. In my lab, we focus on this unique understudied trait of cancer, and we try to take advantage of it in order to selectively kill the cancer cells. Today, I will tell you a bit about aneuploidy and its importance in cancer, and I will share with you an exciting project in which we recently identified new therapeutic targets that are associated with aneuploidy in cancer. This project will soon be published in the journal Nature in 2 weeks' time.
I'm going to interrupt and say this is a really, really high profile paper, and we're very lucky to be hearing from Uri in person about it. Go ahead.
Thank you. So as most of you probably know, our DNA is packaged into chromosomes. These threads are composed of DNA and proteins, and we can stain each chromosome in a different color, as you can see in this beautiful picture, and then order them in order to keep track of their number and their structure. Each cell in our body has exactly 2 sets of 23 chromosomes, one set that we inherited from our father and another set that we inherited from our mother. You all probably heard of the company 23andMe. So the name of this company is coming from the number of chromosomes that we humans have in our cells. So all of the normal cells have the same number of chromosomes, 2 sets of 23, and you can see a representative of a normal cell here on the left. However, in cancer, this number changes. Cancer cells have an abnormal number of chromosomes, sometimes more, sometimes fewer, and this phenomenon is what we call aneuploidy. Here on the right, you can see an example of a typical cervical cancer cell. You can see that it has, for example, 3 copies of chromosome number 1 and only 1 copy of chromosome number 2, and so on. And this is really a striking cellular difference between normal cells and cancer cells. Aneuploidy is considered a paradox in biology, because on the one hand, when it occurs during embryonic development, it is very bad for the cells. Here on the left, you can see that sometimes during developments, cell can acquire an extra copy of chromosome 21. And this is the cause of Down syndrome. Moreover, most of the natural miscarriages during the first trimester are actually caused due to aneuploidy in the developing embryo, demonstrating how important it is for our cells to have the exact number of chromosomes. In contrast, aneuploidy is a hallmark of cancer, and we have evidence to suggest that it can actually be good for cancer cells in the sense that it promotes their growth and their expansion. In cervical cancer, for example, aneuploidy is associated with worse prognosis and increased resistance to anti-cancer drugs. Aneuploidy is therefore an elephant in the room of cancer genetics. We all know that it's important, and in fact, it was first discovered in cancer over a century ago. But at the same time, it's been understudied for many years, and we currently do not have any therapeutic interventions that directly target this phenomenon. So in my lab, we try to study this question and answer several sub-questions. What causes a cell to become aneuploid? What is the role or the multiple roles that aneuploidy plays in cancer formation and progression? And perhaps most importantly, how can we target aneuploid cells? And I will briefly present one such study that we recently completed to address this question. In this study, which will be published in a few weeks, we analyzed the landscape of aneuploidy in about 1,000 distinct cancer cell lines. Cancer cell lines are cancer cells that we can grow on plastic in our lab, and we can profile their chromosomes in order to assess how aneuploid each of them is, which is exactly what we did. We then divided the cells into the most aneuploid and the least aneuploid, and compared their sensitivities to thousands of drugs and genetic perturbations. We found that the degree of aneuploidy varies greatly across tumor types. Some of the cancers that are the hardest to treat, such as pancreatic cancer and ovarian cancer, are actually highly aneuploid. So if we find good ways, specific ways to eliminate aneuploid cells, we may be able to offer some new therapies to these very important cancer types. And we were able to identify several vulnerabilities associated in aneuploidy in these cancer cells. A major Achilles heel that we found is the increased sensitivity of aneuploid cells to inhibition of the mitotic checkpoint, which is an important layer of regulation on cell division. Here on the left, you can see normal cells treated with this inhibitor. The red here is just an artificial color that enables us to see the border of the cells. And what you can see is that this cell culture is mostly red, meaning that the cells are viable and they proliferate in the presence of the drug. And these are cells with a normal number of chromosomes. In contrast, on the right, you can see the aneuploid cells that were treated with the very same drug. There are way fewer cells here, and the ones that are still there are large and in general are very unhappy. So there's a clear difference in the response of normal and aneuploid cells to this particular drug. We therefore characterized the cellular, molecular, and functional consequences of targeting this particular pathway in normal and aneuploid cells. I'm not going to go into the details of this diagram, but suffice to say that we were able to go pretty deeply into the mechanism that leads to this profoundly different drug response that I showed you in the previous slide. It's important for me to say that this project was a...
Can I interrupt with a question about that?
Of course.
So did you look at a specific kind of cancer when you did this, or did you look at different types of cancer cells? How general do you think your results might be?
Yeah, that's a great question, Nancy. So we did a pan-cancer analysis, meaning that we included all cancer types for which we had data. We focused on solid tumors, but this included quite a large array of solid tumors, including breast, lung, ovarian, cervical, pancreatic cancers, and several more. We had about 24 cancer types included in this analysis.
So that's back a couple of slides. Can we go back and look so people can appreciate how much work has gone into it? Because as I recall, you looked at nearly 1,000 different cell lines. Is that right?
Yes. Yes,
wow.
So each cell line is coming from an independent tumor, from a patient's tumor, and it was derived by different labs in the world in order to actually get it to grow on plastic in the lab. But we were fortunate to be able to gather these cell lines and to analyze the genomic features of these cell lines. And the main novelty of our study was that we were able to assign aneuploidy status, or to profile the aneuploidy status of these 1,000 cell lines. So once you have 1,000 samples, you get to enjoy the benefit of the large numbers. And that enables us to study it pretty comprehensively.
So it would be great if you'd walk us through one of the boxes. What are the little dots for, and what's the line in the middle, just so people understand how you take massive amount of data and compress it to something that's useful in spying on biology.
So in this particular plot, each dot is actually a cell line, okay? And we divide them by the tumor type. So you can see there are, I don't know, about 15, 17 tumor types in this particular plot. And the y-axis here is the aneuploidy score. So this is the measure that we determined in order to define how aneuploid each cancer cell line is. So you can see here on the left are the more aneuploid cancer types, and on the right are the least aneuploid cancer types, with the a line that you referred to being the mean of each tumor type. Okay, so basically everything that is on the left part of this plot represents a high degree of aneuploidy. And these are the most interesting tumors from our perspective, because these are the ones that would be more susceptible potentially to whatever we identify as an Achilles heel of aneuploidy.
And so this is interesting, not just because there's some that are potentially very sensitive, but there are also tumors that seem not to be potentially aneuploidy. And those look like tumors of the blood or of the nervous system. Is that correct?
This is correct. Even those are sometimes aneuploid, so it would be incorrect to say that they are completely not aneuploid, but they have a much lower degree of aneuploidy. So fewer chromosomes on average are affected by the phenomenon that I've been describing in these particular tumor types. And you accurately observed that blood tumors and peripheral nervous system tumors are less affected by this phenomenon.
But it would be good news for pancreatic tumors and ovarian tumors and some other very difficult to treat tumors, because they're way over in the high aneuploidy category.
Right, but one thing that I think is important to explain, though, is that we also, for whatever we find, we also need to do the analysis within the tumor type, because when you just compare across tumor types, then the degree of aneuploidy is one potential difference, but there are many other potential differences between different tumors. If you take lung cancer and compare it to breast cancer, then the analysis would be inherently confounded by other parameters. So one other important analysis that we performed was to basically repeat every analysis that we did within each tumor type. So therefore, for any finding that we present, we can say not only that the aneuploid tumor types would be relevant, but also that patients that have more aneuploidy tumors within that particular tumor type would presumably benefit more from a given drug or a given intervention. So even for tumors with, let's say, an intermediate aneuploidy level as a whole, we can identify the specific patients that have more aneuploidy in their tumors, and these are the ones that we would predict to respond better to these particular treatments.
And this is sort of what's known in shorthand as personalized medicine. You look
exactly
at the patient's individual cancer. You don't just say, okay, it's ovarian cancer, we'll do this. Yeah, okay, good. Sorry for the interruption. Keep going.
No, it was an important interruption. Thank you for interrupting. Okay, so I was towards the end, and I just wanted to mention that this project was a successful collaboration between 7 laboratories across 5 countries. And this came particularly useful in the past few months because we were all going into and out of lockdowns and quarantines. So at least at every time point, there was one lab that was open so that we could keep the science going. So, you know, it's another benefit of worldwide collaborations. And I would like to thank my team, and especially Dr. Yael Cohen-Sharir, my lab manager, who's also the first author of the paper that I shared with you. And here you can see our lab meetings and what they look like in these days, which everything is being held via Zoom. And finally, I would like to thank our funding agencies, and in particular the ICRF, for awarding me the Gesher Award for scientists to establish their cancer research lab in Israel. I think that if we ever want to conquer cancer, we must invest in basic cancer research. There's simply no other way. And the generous contributions from donors like those who are in the audience today is absolutely critical for enabling us to do this research and make discoveries that hopefully in the near to far future will be translated into improving patients' clinical care. So thank you, and I'll be happy to address any further questions if you have any questions, Nancy, or anyone else has any questions.
So I'm actually okay with questions. We're recording this webinar in advance because of weather issues in the United States causing some problems with internet in the winter. And we'd really be glad if people who are watching who have questions, send them to ICRF, who will relay them to me and Uri, and possibly to physicians who might be able to comment better if these are medical questions. So we'd love to get questions and comments from our viewers, and we will answer all of them that come in. This is really exciting. I've completely enjoyed getting to know you and to hear about your science, Uri, and you couldn't have made a better case for supporting basic research. So thank you for doing that too.
Thank you, Nancy. The pleasure was mine.
Oh, thank you. So to our wonderful audience, thank you for watching. Our next webinars will feature first in February, Iri Ben Aharon at Rambam, who'll talk about cancer and pregnancy, which is a really, really important issue. And then after that, Shamgar Ben Eliyahu at Tel Aviv University, like Iri, who's going to talk about new pre and post-operative treatments that can advance cancer therapies. You can learn more about them and their talks on the ICRF website. And till then, have a good winter, and we hope to see you again in the future. Thank you.